So now let's look at the mismatch repair in E. Coli. So this particular video, we are looking at mismatch repair in E. Coli. Mismatch repair in E. Coli. So there are a few things we need to understand before we start. So one is, what is this mismatch repair? So mismatch repair is a case where we have those errors that escape the proofreading. Remember, when we are having our replication, we have what we call proofreading. So all errors that escape the proofreading part will be corrected by the mismatch repair. And this particular video, we are looking at mismatch repair in E. Coli, which is totally different from that of eukaryotes, as we shall see. So we can define it as the post-replication correction of errors. You get? So that is mismatch repair. Then another thing, the enzymes that are involved. So in mismatch repair, we have a variety of enzymes which are called mute proteins. We have mute proteins. So we have protein, S, we have S, we have L, and we have H proteins. Then the other ones will be helicase. We shall have helicase helping us at one particular point. We shall have exonucleases, which will depend on what will be damaged. So nucleases, and then shall have polymerase, that is DNA polymerase type 3 specifically, and then shall have ligase. So these are like the main enzymes, but not forgetting we also have what we call a DAM methylase. So we are going to look at the importance of each. So another point we need to take note of is the steps involved. So there are about personally I've found about like five main steps, and then we must know the principle on which it works. So the principle in which it works by recognizing unmethylated strands, unmethylated DNA strand. So you will find that we have, after replication, we shall have the template DNA strands, or the parental DNA, parental, and then shall have the daughter, that is what's formed, the daughter DNA. So must know that errors are not going to occur in the parental DNA because it's what you are copying. So we expect to have errors in this daughter DNA. Could be an insertion, could be a deletion, such errors. So the errors are going to be in the daughter DNA. So what will happen is that this DAM methylase here, the one we've just seen, it will look for a particular site on the parental DNA, which is known as, it will be a sequence containing, it will look for that sequence, which is known as GATC, guanine adenine thymine cytosine sequence. It will look for that sequence on the parental DNA, and it will fully methylate this region it will add those methyl groups, that is methylation. So the DAM methylase, it will come and methylate, but what specifically? It will methylate the adenine nucleotides. The DAM in full is DNA adenine methylase, you get? So that's why we have the DAM. Adenine specifically is what will be methylated, you get? So it will methylate the adenine nucleotides uh, in the guide sequence of the parental DNA strands. You get? So this will help us recognize only the unmethylated, and we shall have the unmethylated in the daughter DNA. So this one will have unmethylated, unmethylated uh, GAT-C sequences. So these are what uh, this is what is going to be recognized by the mute proteins, the GAT-C, which is unmethylated. So that's like the principle on which it works. So we have seen the function of the methylase, which will come in to methylate the GAT-C, specifically the adenine nucleotides within the GAT-C sequences, which are found in the parental strands. So what will happen is that we have our daughter DNA and its uh, strands. So what will happen is that we have S, L, and H. These will be the proteins that will initiate this process. So we have S, which will scan. We have mute L, which will link, and H, which will create a nick or cut. So S will be the one to come in first. So we shall have mute S, which will come and scan the uh, the DNA of this daughter, the daughter DNA for the mismatch. So how will it do it? It will do it in an ATP dependent manner, meaning it will require some ATP to slide along these strands. You get? So it will require some ATP, and it itself has an ATP ATPase activity in that it will hydrolyze this ATP. You get? So after activating or finding the mismatch, it will recruit this mute S now. Mute S will come in and they will form a complex. You get? But remember, all this requires ATP. So they will form a complex which is known as the mute S, mute S, mute S, mute L DNA complex. Remember, they are bound to DNA. Mute L DNA complex. So after forming this complex, what we shall see is that this complex itself, after locating these sites, the GATC, it will locate the mismatch in short. So locating the mismatch, I think I'll need to extend my board for clear view. Don't forget our enzymes that we had. So after locating the mismatch, let's say it's somewhere here. So what will happen is that this complex will activate the mute H. But upon locating the mismatch, it will activate the mute H. So hope you've seen the sequence that we have mute S first, mute L comes in, recruited by S, then this complex will recruit H. So H will come and create a nick. Why is it creating the nick? The nick will be created either up or downstream of the area of the mismatch. But remember, the area that's recognized is what? GAT-C. You get? The unmethylated region. But this one will come in and create a niche around upstream or downstream of the mismatch. Let's say here, downstream. So it will create a niche downstream of the mismatch. So where are we having the other complex? The other complex, for it will be like on the opposite side. You get? We are going to see why it's going to be on the opposite side. It will aid in locating where these exonucleases are going to stop. So we have seen that we have created a niche, and what will happen next, we shall have our helicase now coming in. Remember, helicase is for unwinding. So helicase will come and unwind the strands so that we have two separate strands created. But remember, this one has a nick or cut somewhere near the mismatch. Let me say this is our region of mismatch. So helicase has unwound the two strands. So we shall have another strand which is normal, having the single strand binding proteins coming in to stabilize it and prevent it from forming secondary structures. Whereas this one, which is has the mismatch, will be acted on by the exonucleases. So remember, exonucleases require a free end to do the activity of degradation. That's why we create the nick, you get, so that we create a free end and it will break or degrade this DNA towards the mismatch. So this is where we have to look at the types of the exonucleases now. So if we have our strand, let's say we have this side as the 5 prime, this side the 3 prime. So exonucleases also have types. There are those that have the 3 prime to 5 prime exonuclease activity. For example, exonuclease 1, exonuclease 1. Then there are those that have 5 prime to 3 prime exonuclease activity. For example, exonuclease 7, exonuclease 7, or there is another one which is called RECJ. So this RECJ, some will tell you that it's a bidirectional. It can go in both directions. So if the mismatch is here and we have the other complex, don't forget the complex is somewhere here to indicate where to stop. You get? So the exonuclease we are going to have will depend on the direction of the nick and the mismatch. So you can see that the nick is in the 5 prime end, whereas the mismatch is in the 3 prime end. So we'll need some the exonuclease which can move from the 5 prime towards the 3 prime. And which one will that be? It will be exonuclease 7. So it will be recruited here and it will cut and up to the when it reaches this complex signaling the stop, but it will degrade everything here. If the mismatch was in the 3 prime end, let's say if it was this side, I mean the 5 prime end, then we would recruit something or exonuclease which moves from the 3 prime towards the 5 prime. Start from here, degrades everything until it reaches that complex signaling the stop. So after we have degraded this, we need to now replace with the correct enzymes. So that's when we are going to have our polymerase now coming in to synthesize or replace with the correct nucleotides. That's when we have our polymerase coming in, and after replacing it with the correct nucleotides, then we have our ligase which can fill in any nicks that are created, and that's how we have our mismatch simplified in short. Thank you for this video. We shall have more about this biochemistry. Thank you for watching.